Hello and welcome back to my YouTube channel. Today we are going to discuss about meiosis. Now meiosis is a reductional division, and why it is known as reductional division? Because the parent cell with 2n number of chromosome produces the daughter cells with n number of chromosome or half number of chromosome, that is the reduced number of chromosome. Now in meiosis there are gametes produced in the case of humans and animals. And in case of plants, pollen grains and ovules are produced. Now, before going deep into the discussion of meiosis, let us first discuss about the cell cycle. Now, the cell cycle is divided into two phases. The first phase is the interphase, which is also known as the resting phase, although it is not a resting phase because the active synthesis of RNA, proteins, and DNA takes place over here. So, in G1 phase of interphase, the synthesis of RNA and proteins along with the active synthesis of cytoplasm take place. Now the second part is S phase, that is known as synthesis phase. The DNA duplication takes place in this phase. And the third one being G2 phase, in this the synthesis of RNA and proteins takes place. Now, after synthesizing the effective machinery, the cell enters into a phase that is M phase or meiosis. Now, is of two stages. That is the first stage being meiosis I, that is heterotypic division. Why it is known as heterotypic? Because hetero means different and typic means type. So different type of cells are produced than the parent cell. That is 2n cell, that is the parent cell, will produce the daughter cells with half the number of chromosome or different number of chromosomes. Now meiosis II is known as homotypic division. That means homo means same and typic means type. So same types of cell produced due to cell division is called homotypic division or meiosis II. Now meiosis I is divided into four phases. These are Prophase I, Metaphase I, Anaphase I and Telophase I. Now in Prophase I we have five stages. First one being leptotene. In leptotene, we find that the chromosome become uncoiled and appear thread-like. So there are thread-like chromosomes appearing. Then there are centrioles duplicating and moving towards the opposite poles. After this, the cell enters into the zygotene stage of prophase I. In zygotene, the pairing of homologous chromosomes called synapsis takes place. And synapsis may occur at one or more point along the homologous chromosome. This leads to the formation of synaptonymal complex at the point of the formation of synapse. After this, the cell enters into the another stage of prophase I, that is pachytene. In pachytene, we will find that the chromosome become paired and twisted and they cannot be distinguished. In this stage, we find that the crossing over taking place at the different points in chromosome. This involves mutual exchange of genetic material and therefore it leads to variation amongst the generation. That means the biological parents are different from their children and the biological siblings are different from each other. After this stage, the cell enters into diploid stage. Now the chromosome starts moving far apart. During this separation, the homologous chromosome forms bivalent and this is seen attached with each other at one or more point Like you can see over here, the chromosomes are being attached at different points, forming chiasma. This is known as chiasma. Now, after this, the cell enters into another stage known as dikinesis. Now, chiasma moves towards the end of the chromosome and leads to chiasma terminalization. Over here, you can see the chiasma terminalization taking place. After this, the cell enters into the metaphase I. Here, in metaphase, you can find the chromosome aligned in the equatorial plane. Homologous chromosome aligned the equatorial plane and attaches to the spindle fiber through the centromere. After metaphase, the cell enters into anaphase. Now, in anaphase, you'll find the chromosome moving towards the opposite poles. The sister chromatids still remain attached together in anaphase I and separate out in the second meiotic division, that is, during anaphase II. The separation of homologous chromosome during meiosis is known as disjunction. After this, the cell enters into telophase. In anaphase only, we see the cleavage has started being formed and it is centripetal type of cytokinesis. Now, after this anaphase 1, the cell enters into telophase where the nuclear membrane is reappearing. Nuclear membrane is reappearing and the cell divides by cytokinesis. So the division of cytoplasm has taken place. The daughter cells receives haploid number of chromosome. After this, the cell enters into meiosis division 2. In that, we will see that it is just like mitosis, but no DNA replication takes place over here, and there is division of haploid chromosome over here. In the case of mitosis, it was diploid, but here haploid chromosome are going to be divided. Now in Prophase II, we find the centrioles are going to migrate towards the opposite poles and spindle fiber starts being formed between the two opposite poles. After this, the cell enters into metaphase II. In metaphase II, the chromosome aligned at equatorial plane. The chromosome aligned at equatorial plane and then they get attached to the spindle fibers through the centromere. You can see they're aligned on the equatorial plane. After this, the cell enters into anaphase 2. In anaphase 2, again, the chromosome move towards the opposite poles and the cytokinesis begins. After that, the cell enters into telophase where the cytokinesis has taken place and the nuclear membrane reappears, nucleolus reappears, and therefore this ends meiosis II. Now this I think so this meiosis is clear to you all and you find my video informative. So do like and subscribe my channel and hit the like button and a notification button in order to get the latest notification of the